This program is a presentation of UCTV for educational and non-commercial use only.
I'm here to talk about how forces help organize and regulate biological systems. So we've heard a lot about very different manifestations of the concepts of systems biology and what exactly that means. For me, I'm very interested in understanding how cells are organized, how they're put together, how they get their strength, how they move, how they divide. And as you can imagine, that requires the coordinated action of literally tens of thousands of proteins, some of which are responsible for chemical signaling, some of which are responsible for mechanical signaling. And I'm really interested in that interface, which is how do cells recognize chemical signals and then convert those chemical signals into force and into mechanics. So the length scales, just to sort of set your expectations, the length scales that I'm interested in are from that of single proteins, which are several nanometers, up to whole cells, which are hundreds of microns. And I'm interested in how cells grow, but I'm not necessarily counting their number in large quantity. So that's sort of the length scale that I'm interested in. So I'd like to start by giving you several examples that I think are illustrative of the role of force in biology. So I think a really nice example is one of cell motility. So motility is movement, right? This is how cells move from one area to another. And in order for cells to move, they have to all of the proteins inside have to sort of collectively decide, I no longer want to be here, I want to be over there. And then they have to collectively move so that they get from point A to point B. And one of the ways that happens is by structural proteins, which are inside the cell, forming larger networks at one side and dissolving those networks at the other. And the networks are literally pushing the membrane forward and causing this motion. And then the retraction occurs when those networks dissolve. So these are scaffold proteins whose job is to really exert force and move in cells. Now, the diagram that's here, which is I grabbed from an NIH website, shows you not scaffold proteins, but a different type of network. And it's the network that we heard about this morning and a network that a lot of the previous speakers spoke about. And it's sort of a chemical network or a signaling network that has to do with interactions of specific molecules or interactions of specific proteins. And this is clearly important to cell motility, but there's another network that's involved. It's sort of a shadow network, which is the mechanical network that's actually holding everything together. And I think that interface is a really interesting one and one that we're just beginning to scratch the surface of. So how is it that cells can recognize force and recognize tension? Experiments have been done that show that if you put motile cells down on substrates that have a variable elasticity, they'll crawl towards the stiffest part. So somehow they recognize their substrate, and they recognize the mechanical properties of their substrate. If you were to attach a small probe, a mechanical probe to the cell and pull, you'd find that it took a certain amount of force to pull the first time, and then if you pull it again, it's a lot harder. It's a lot harder to pull, and that's because the network has responded to that force. So I'd like to give you some intuition about how those sorts of forces are felt and how that force signal is converted into chemical signaling in cells. One way is actually through the plasma membrane. So this is an electron micrograph of a membrane. This happens to be a red blood cell membrane, but there are membranes on all animal cells. They're important for containing and organizing the contents of the cell. And the major component is really lipids, which are organizing the cell and providing an envelope for everything that's inside. But a key component are these transmembrane proteins, which are large enough to embed into the network and actually, in some cases, form channels from the inside to out. One particularly important type of channel is what's called a mechanosensitive channel. And this is sensitive to stretch or bending of the membrane and can actually open or close depending upon the force that it locally feels. So this is one example of a force-sensitive switch that opens or closes, allowing the movement of specific ions, in this case, through the channel based entirely on force and tension. So this is part of this story. These types of channels are part of the story for cell motility. It's only part, I think. My work actually focuses on a different part of the story, which is how these networks of polymers are organized and how actually motor proteins, which are small proteins that can go through conformational changes, restructure those mechanical networks and exert force. Clearly, these are all interconnected. And for those of you who maybe before this session hadn't had too much exposure to the concept of networks and to systems biology, I want to apologize in advance for my next statement, but these are not nearly complicated enough. So whenever you look, or whenever any of us look at a new signaling diagram or a new interaction map, it always takes a while to get your bearings. They're very complicated. There's a lot of proteins here. They have funny names. And I would argue that in addition to these kind of connections between specific chemical interactions, we really need to include force. We need springs. We need a way to understand where tension is acting and how it's causing specific types of changes in cells. And in particular, I think we need to develop techniques that allow us to visualize force and visualize strain in cells in the same way that we can visualize stuff. So we know where all of these things are, in large part because we can label them fluorescently and watch them. I think that we need, as a group, to understand how forces are generated in cells and how we can do those types of measurements. So motile cells are really only one example of cells that are sensitive to force. Another example, which I think is really becoming more and more prevalent, are stem cells and their ability to differentiate. So stem cells, as you know, have the potential to develop into many different types of cells in the body, and they're of course, a very exciting new type of cell biology which is emerging, which is giving great promise to different types of regenerative medicine. I've shown in cartoon form here the idea that a single stem cell can differentiate into several different cell types, right? Skin cells, muscle cells, blood cells. And it's interesting to sort of wonder what causes this type of differentiation, what signals that when the cells are put in an organism or put in a petri dish. And I think a really interesting study recently, which was published in Cell by Engler, shows that in addition to all of the chemical specificity that we would kind of imagine, mechanical substrate elasticity is also extremely important to how these cells behave. So stem cells, which were plated onto substrates of different stiffness, but held in entirely identical chemical conditions, were shown to differentiate specifically due to substrate stiffness. So if you put them on very soft substrates, they differentiated into neurons. If you put them on substrates of intermediate stiffness, they differentiated into muscles. And if you put them on substrates of high stiffness, they differentiated into bone. And here, neurons are soft, muscles sort of intermediately soft, and bone is very hard. So even in identical chemical conditions, you can see that you can drive differentiation purely by mechanical means. And I think that this is important for a few reasons. One, it's beginning to tell us some really interesting information about stem cells and how they're regulated. But additionally, it's telling us, cells pay attention to their environment. Maybe stem cells are kind of an extreme example of this. But I think that this is going to be true of a lot of cell biology. And that I think we have to be careful when we look at cells and when we look at networks to make sure that their mechanical environment is as tailored as their chemical environment is. Another example of force sensitivity in biology has to do with cell division. So this is a little bit different because the past two examples I showed you really have to do with cells recognizing and adapting to changes in their environment which are external to the cell. This is looking at changes in the environment which is internal to a cell. So this is a picture of a mitotic spindle. Here we have chromosomes, which are labeled with a blue fluorescent dye, and microtubules, which are a particular type of biopolymer, which are labeled with a green fluorescent dye. You can kind of imagine that this is a large cell on the outside, and these chromosomes have lined up in the middle. And the whole job of cell division is to separate out the chromosomes into two daughter cells. So each daughter has an identical copy of the genetic code so that that daughter can go off and live and be happy and have all the proteins that it needs. So the job of this machine is actually really critical to cell health and to organism health. And the chromosomes need to be exactly ordered in the right way, and they need to be exactly separated in the right way in order for healthy tissue development and healthy organism development. Once everything is aligned and everything's ready to go, there's a separation that occurs, and you can see the DNA now being brought to the two corners of the cell. A pinch is going to happen in a minute here that pinches off the two daughters. This process is known to be very tension sensitive. There are in fact several feedback mechanisms at this stage that make sure that this division does not occur until the spindle has the proper structure and all the microtubules are under the proper tension. So I think that we see in time and time again, and example after example, that we cannot consider force and mechanical regulation independently of other types of cell biological processes. We really need an integrated approach. So my lab is in particular interested in cell division and the process by which this occurs, and I'd like to now kind of move away from specific sort of overview examples to specific work that's done in my laboratory. We're interested in lots of things. One of the things we're interested in is biopolymer networks. And again, I use network here to mean something a little bit different. I mean network to mean a physical network, things that are physically connected, by the most part, by these biopolymers. Here, these are microtubules, again, which are shown in cells that are not dividing, but rather are spread. We're interested in whole cell mechanics, how cells get their strength, how they move, how they divide. We're interested in cellular machines and understanding how a machine like the spindle works, how it's robust. And then, of course, we're interested in the molecular generators of motion, which are these motor proteins that can convert chemical energy into force and movement on the nanoscale. So some of our primary questions are, how do these protein-based machines work? How do nanoscale proteins generate large-scale motions? So if most of the force generation is happening because polymers are growing in nanometer increments or very small proteins are doing movements, how are they coordinated at the systems level to allow for division or to allow for development? How do cells generate and respond to forces? What determines strength? And then can we mimic nature to make new types of machines? So one of the things we're most interested in are these nanoscale motors. This is a cartoon which was developed by HHMI. And this is a typical shape of one of the motors that we're most interested in called Kinesin. So Kinesin has basically 2 feet and 2 arms. And its feet move along polymers, which act as roadways, and its arms hold onto cargo. So Kinesin's job in the cell is kind of like your FedEx truck. It moves along a highway, and it totes stuff from one place to another. And we're very interested in understanding how this happens. These are the primary generators, or motor proteins generally, not just kinesins, but motor proteins generally are the primary generators of force and motion in cells. They're responsible for muscle action. They're responsible for transport neurons and things like sperm motility. They're extremely efficient motors. So just from the perspective, if we sort of divorce ourselves from biology for a moment and just think about engines and energy conversion, they're extremely efficient. So just from the perspective of wanting to build better machines, it's really interesting to study these types of motors. Just to give you a sense of the length scales and time scales, the step size here is 8 nanometers. It's actually specified by the polymer structure. The speeds are up to 100 steps per second. So imagine if you were to be able to run that fast. I mean, these book, right? So they can sustain loads of about 7 piconewtons, which requires conversion of about 60% of the energy that they use. And they can take roughly 100 steps before detaching. So we're interested in understanding the molecular mechanism of these systems. Of course, they're essential to human health. If you have misregulation of kinesins, you will inevitably cause disease. So our research goals are to identify protein modules that are responsible for specific behaviors, then generate novel motor proteins using bacterial expression, and use those engineered proteins to make novel nanoscale machines. And these are really our engineering perspectives, but it gives us a sense of being able to connect specific parts of this protein to specific behaviors. And our goal is, if we can understand those behaviors and we can modulate those behaviors, then we can understand how these proteins act in the context of larger assemblies and act in the context of larger systems. So I sort of showed you that kinesin protein as if it was a singular entity and there was only one. It turns out there's not. It turns out there's lots of them. So we have quite a lot of natural, I guess, models to work from. There are over 50 kinesin genes that are essential in humans. There are far more if you start to include other organisms. We are interested in characterizing the mechanics of these naturally occurring proteins and then comparing structures, kind of mixing and matching novel sequences to create hybrid motors. I should say that most of these are not well understood and they're not characterized. We heard this morning about a systems approach to medicine where you're actually able to take proteins from individual patients and try to understand the differences in those proteins on a human-to-human individual basis. I mean, we're talking here about, we don't have any proteins in some cases from any organism that we understand. So trying to bring mechanics into a systems level diagnosis from medicine, I don't think we're at that point yet. But I think that it's really important to consider the role of mechanics and elasticity in systems levels responses. So motors both generate and are regulated by force, and that's true, I think, of cells in general. So it's sort of an interesting system because it's able to actually create force, and then in creating force, regulate itself, because some of the biochemical rates are actually force dependent. So this is a crystal structure of this part of the kinesin, and the crystal structure was solved in the absence of the microtubule. And you can see the distance between the two motor heads is 5 nanometers. The distance when these two heads are both bound to the microtubule is 8 nanometers, which means that there's really significant strain put on the motor when it's attached to the microtubule. That strain is really important to regulating the velocity to which these motors move. So we understand that force is important even at this single molecule level. To really be able to better understand these force dependencies, it's important for us to be able to visualize single molecules and then apply force in a controllable way and see what that force does. So in order to visualize molecules, we use recombinant DNA technologies to be able to express kinesins that bind to a green fluorescent protein, and we do this through recombinant techniques, so they're expressed in fusion. So we've removed the normal cargo, I guess, and replaced it with green fluorescent protein. And we can then watch these move along microtubules in cells. And I think one of the really exciting things here is that we can take this DNA, we can put it into bacteria, and we can use the bacteria as factories to make whatever kind of protein we want. So we can use a native kinesin, we can use a modulated kinesin. You notice probably that my appointment is through the Department of Mechanical Engineering. So the students that I have working in my lab don't necessarily come with fantastic or at least intense biology backgrounds. It's quite common for them not to have any biology experience from high school. And we're really lucky at Santa Barbara, and we're really lucky in the lab, in the building where my lab is located, which is the California Nanosystems Institute in Ealing's Hall, that there are core facilities that are particularly built to help this. So we have core facilities for engineers that are fully staffed by biologists, and they provide all of the equipment and all of the training that's needed to do recombinant DNA expression, recombinant DNA work and protein expression in bacteria and also mammalian cells. So the energy barrier to get into this type of work for people who are not biologists per se is really low. So I think that that's a great feature of Santa Barbara, and I think it enables this type of research. So if we look at a microtubule, which is not labeled, but sort of there's one here and there's one here, we can do sparse labeling of kinesins that are GFP labeled. We can look at the signal and we can verify that, that in fact the photobleaching, which is a irreparable damage to the fluorophore, happens in one signal. So we go from signal to background in a single step, which verifies that we can actually visualize single GFP kinesins using microscopy. And we've done this using a custom-built turf microscope. And we're in the process of now using this type of system to actually characterize the activities of different kinesins in vitro. A second technology that we use is a way to apply force. So here we have a kinesin motor. Instead of having an attachment of GFP, we attach it with a small colloidal beads. This is a polymeric bead that has an index of refraction, which is different than that of water. We use a technology called an optical trap, which is a highly focused laser beam. The strong electric field at the area of the focus exerts a force on the dielectric particle and allows us to manipulate that particle in 3 dimensions. So we can not only manipulate the motor protein and bring it to the microtubule, we can also apply forces to the kinesin as it walks. This allows really ultra-high precision measurements. We can apply forces in the range of half to 50 piconewtons, and we can measure positions to roughly angstrom level, which is really crystallographic. I mean, this is, we're able to do incredible position determination with this type of device. And these instruments, unfortunately, are not available commercially, so we design and build them ourselves. One of the nice features is that the particles experience this attractive force back to the center of the laser beam. And for very small displacements, the laser beam can effectively be modeled as a spring. So if you measure the movements of the bead just through thermal motion, you can extract the spring constant. So anytime you know the distance that your particle is moved from the center of your laser beam, you know your force. So it's an easily calibrated system, and we can actually use this to apply forces to motor proteins in really controlled ways. So this is a data record that shows the position of the bead, which is really the position of the kinesin, moving along a microtubule, and you can see the step-like motion occurs. So we're actually able to visualize the individual translocations of the kinesin as it moves along its microtubule substrate. You can see the bead position and the trap position are offset, and they're offset by a constant amount, and that allows us to apply a constant force. And we can easily measure the displacements, which are 8 nanometers here, and if we look at the standard error on the mean, we're well within a nanometer. So one of the things that we're interested in, just to come back here for a second, one of the things we're interested in is, what sets this timing? What sets the timing of that step? How much of it is it set by biochemistry, and how much of it is set by force? So we're in the process of actually screening through a large number of kinesins to be able to better understand what sets the velocity and what sets the waiting time, and then how do we use that to model systems of motor proteins that are working together. Another key research topic in my lab is looking at the mechanical properties of biopolymers themselves. So in the previous work, we really thought of the biopolymer network as kind of a highway upon which the motor proteins moved. In this case, we're actually interested in the properties of the highway. Okay. Microtubules are really interesting polymers. They're formed through a complicated aggregation process or polymerization process where you have growth not only of a single filament, but you have filaments that interact together to form a tube. So microtubules are actually tubes. Their outer diameter is about 25 nanometers. Their inner diameter is 12 nanometers or so. They're hollow. And that gives rise to really interesting properties. Each monomer has lots of lateral connections to the monomers around it. So that makes them very stable, and it makes them very stiff. So another interesting property of these filaments is that although the lateral connections improve their bonding and improve their desire to kind of stay with their neighbors, there's also some chemistry involved. So you can see this color shift from red to kind of pinkish as you go away from the growing tip, and that's because each of these monomers also carries a chemical fuel, and there's a hydrolysis reaction that occurs within the polymer. After hydrolysis, the polymer actually becomes much less stable. So there's a competition between addition of new monomers and complete disassembly of the microtubule. So because this is catalytically active and because it's such a big polymer, it is really interesting and non-intuitive polymerization kinetics and also stiffness measurements. So sort of the origins of stiffness are really different in microtubules than they are in a lot of other kinds of polymers. So we're interested in polymer networks, both in isolation, in adherent cells. Here the microtubules are shown in green, and in nerves. So microtubules form this long process, or form the body of this long process of the nerve axon. So we're interested in understanding how individual microtubules and microtubules working together with other microtubules create the variety of shapes and structures that we see biologically. And then, of course, we're interested in understanding how microtubules are important to dividing cells. So there are a lot of helper proteins that bind onto microtubules, change their properties, change their polymerization rates, and we would argue change their stiffness. And we're in the process now of going through a large series of microtubule associating proteins that are not motor proteins, but rather binding proteins, and understanding how they change filament stiffness on a single polymer level. We do that by taking pictures of microtubules that are confined to 2 dimensions by making extremely thin sample chambers, and we look at their shape. So you can kind of imagine if you had something which was fairly flexible, like a soft piece of yarn or soft piece of plastic tubing, I could bend it pretty easily. I could push on the ends, I could bend the sides, and you'd see like a lot of different shapes that came about. And if we went to increasingly stiff materials and eventually got to something like steel, with that same amount of force, I'd be able to bend it very little. So that gives you some intuition for the idea that shape changes under constant force tell you something about stiffness. So that's really the basic physics behind what we're doing here. We don't apply a force externally. We allow thermal forces to do the work. So we just keep these at room temperature, and thermal forces are enough actually to bend these polymers gently, and then we look at their shape. Stiffer polymers will bend less, obviously. And we use actually a Fourier analysis to be able to look at mode amplitudes and relate mode amplitudes to thermal energy, and then use that to extract out stiffness. And we're interested in doing this both for bare microtubules and also for microtubules that are coated with regulatory proteins. So in addition to looking at single microtubule networks, we're also interested in looking at single microtubules. We're also interested in looking at networks. And I think one of the main driving hypotheses behind this work is trying to understand how force gradients and strain gradients may influence regulation and influence biological processes in the same way that chemical gradients do. So I would argue that force gradients and strain gradients carry information. And if we understand the properties of networks that optimize the transmission of stress and the transmission of force, then we will have a much better understanding of how that force might be converted into chemical signaling or might be converted into electrical signaling. So we look both at networks that are in living cells. This picture is actually from a colleague of mine at Harvard. But we look at both at pictures of microtubule networks in living cells and also isolated networks, and we try to understand how is stress transmitted, what's the optimal network design, and can we eventually use engineered biopolymers to make novel materials. One of the techniques that we're using here is actually a magnetic tweezer technique that's been developed by Omar Saleh's group, and this work has been done in collaboration with one of his graduate students, Jun Lin. And the idea here is to be able to take microtubule networks, embed small magnetic particles, and apply forces, point forces to the network, and try to understand what's the force that's involved in stretching the network, and also by looking at some fiducial marks in the network, rather, whether that be the network itself or small latex beads which are embedded in the network and immune to the magnetic field. Can we measure the elasticity? So we can look at the deformation as a function of time by this process. We can look at the shear modulus, both for entangled networks and cross-link networks. We can see that not only the presence of the crosslinker, but the nature of the crosslinker is really important. So we're in the process of trying to really use engineering principles to understand the molecular basis of stress transmission. And I think I've already sort of talked about this, but biologically important crosslinkers, I think, are the next step. So how does crosslinking affect microstructure and mechanical response? And how do motor proteins and microtubule-associated proteins work in conjunction to be able to generate and recognize mechanical signaling? One of the things that we've just started is using soft lithography techniques to do microcontact printing of proteins onto cover glasses, and we plan to use this to be able to generate microtubule networks that have specific structure. So we can actually control the architecture and test some of our theories about how these mechanical networks interact. And then, the ultimate goal of this work, I think, is to really reconstitute the machinery of cell division and understand how this very complicated process happens. So this is a plant cell that has very large chromosomes, so it's very sort of striking in its division. But we're trying to understand how forces are generated and regulated here, and I think really to be able to understand the system, it's not just about force generation, it's about how force generation feeds back to chemical signaling and how chemical signaling influences the forces that are generated. So although I think our primary focus currently is trying to understand the mechanical properties of these systems, there's no way to understand this without getting the chemistry involved, and I would say that's vice versa for biochemists who are looking at these types of networks. And then this is some key collaborators in our group, and also the students that I'm working with and our funders. So thank you. The question was, what about development and the idea of cells moving and then deciding to stop? There's certainly plenty of work on that front. It's not work that we're doing within my lab. But I think that, I think that it falls into the same category of systems where we need to consider both mechanical properties as well as biochemical properties when we think about development. But.
I realize this is a conference on systems biology, but I got to say that I came into this very skeptical, as most biologists are still. That's a fact. And that's because systems biology still has not, or systems engineering of biological systems, I should more accurately say, has not really been able to be very predictive. Despite the fact that there are claims that it's predictive, I think what I'd like to make the case with the system I'm going to tell you about in simple bacteria, that even a simple system such as an epigenetic switch that I'm going to talk about has incredible complexity. We've been studying, my lab's been studying one small region of DNA, about a kilobase pair, which is only 1,000 bases, for 15 years for this project. And the more we study, the more we realize that there's more to know. So studying really complex systems, you can still get a lot out of it, but I'd like to make the case that you need to first understand a simple system. And we're not quite there yet, but I am now coming around and actually believing that systems engineers can do something for me as a biologist. In fact, I'm convinced of it, and that's why I'm participating. So I didn't come willingly. I came because our system was incredibly complex and too complex for us to understand. We weren't smart enough to figure it out. And so we started, let's see if I can go back. We started by collaborating with Mustafa Kamash in engineering, and at the time his student Brian Munsky, who's now at Los Alamos. As well, I got Rick Dahlquist and John Perona interested in the structures of the regulators that I'm going to talk about. And one that I'll mention at the end, Mike Mahan's lab got interested in the pathogenesis aspects and actually has made a lot of progress in understanding how the epigenetic system regulates virulence. So the system is the PAP stands for pyelonephritis associated pili. Pyelonephritis is pussy kidney. So if you're a woman and you get pyelonephritis, and women are by and large the major population to get infected, that means that the bacteria is able to get into your bladder and crawl up your ureters into your kidney. And one way that they do it is via these pili, which are marked with colloidal gold on this left bacterium. These right bacteria, which are clonal, they're from the same clone, do not express. These large guys are flagella that allow them to move and swim up the bladder, actually swim up the ureters. And as we affectionately say, swim up the ureters to find kidney pie, which for them is their food. So they can go up to the kidneys. So you'll notice that there's a switch. These bacteria are making the pili. These bacteria are not. We noticed this about 20 years ago and have tried to figure out how that works. One of the ways as geneticists that we can do that is by fusing the genes to beta-galactosidase, which is simply the LAC-Z gene. And on indicator media, we can see on cells are blue and off cells are white. And for many years, this is the way that we would analyze switching and switch rates. So the switch rate from off to on, which we'll be talking about primarily in this talk, is 1 out of 10,000 per cell per generation, which is very low. The switch from on to off is 100-fold higher at 1 out of 100 per cell per generation. Currently, we use fluorescence-activated cell sorting with green fluorescent reporters to look at the switch, but this is still very valuable as a genetic tool. So the organization of the PAP operon, so the entire function of this operon is really to make a pilus, which is shown right here, and the pilus consists of a rigid base made of a PAP-A subunit and a flexible tip made of a number of subunits. And this allows them to attach to host cells, for example, bladder cells. And there are many other switches with this type of organization that everything I'm going to talk about relates to. Over 30 different types of pili systems and many different pathogens. And we're going to focus mostly on this region here with the PAP-I regulatory protein, this 1 kilobase pair or so intergenic region and PAP-B, with the promoter, which is allowing transcription of this entire region to allow pili to be made. The structures, as I said, for LRP, which is one of the regulators that we discovered or co-discovered in my lab many years ago, called leucine responsive regulatory protein. It forms an octamer, and John Perona's lab has shown that it forms an octamer and has the structures. And recently, Rick Dahlquist's lab has 2D NMR structure of PAP-I, which is the co-regulator. So you're going to find out that PAP-I and LRP are the main regulators of this switch, in addition to a methylase, because it's an epigenetic switch. And I'll remind you that epigenetics, what is an epigenetic switch? Epigenetic means that it doesn't involve an actual mutation or a rearrangement or change in the DNA sequence. So in this case, we're going to talk about a methyl group, which can be put on a GATSI site, which Martin Marinis, who really started a lot of this field, calls the great GATSI. So you need a little humor in this. So the great GATSI, okay? So now the Pap regulatory region, I told you that we'd focus on the Pap regulatory region, and the way the bacteria have arranged this is very logical, which is nice. There are 2 sets of 3 binding sites. 1, 2, 3 are listed in red because we're going to find that when LRP binds to these 3 sites cooperatively, it blocks transcription, the promoter's right here, and it blocks transcription by binding right where the promoter is. The regions that are called -35 and -10, where the sigma 70 factor of RNA polymerase binds, are going to be occluded by LRP binding here. Also notice when LRP binds here, it's going to protect this GATSI site from methylation. That's a real key to this switch. And upstream, we've got the same kind of organization. 3 sites, green. When LRP binds to these sites, we're going to switch on. And again, we have a GATSI site, a second GATSI site, a distal site, located in site 5, so that binding of LRP cooperatively to these 3 sites will cause you to turn on. That, in addition, requires some other factors that I'm not going to tell you about, which include cyclic AMP catabolite gene activator protein. So here we have a region where LRP is bound and we're off. We're in the off phase, and we're also going to find that that blocks methylation. And to go on, we need to move LRP from this set of sites to this set of sites, which sounds very simple, but it isn't quite that simple. And we'll be talking about that really the entire time. So DNA methylation comes into the switch because the 2 GATSI sites are targets for DNA adenine methylase, or DAM. So you can talk about DAM this and DAM that, and it's okay, because we're going to be talking about DAM. And DAM has the ability to methylate GATC sites, except if they are bound by proteins such as LRP. So notice, in the off phase, when we have LRP bound to the sets of sites that are proximal to the promoter, and we're off, look what happens to the methylation state. We have an unmethylated GATSI site. And my lab discovered that unmethylated GATSI sites are patterns, and I think we were the first lab to show that these patterns directly control gene expression. Now we know that in eukaryotes, these patterns control many different activities, including transcription. Even brain activity is controlled by DNA methylation in mammals. In the on phase, LRP moves, as I said, to the upstream set of sites and creates a different pattern. Now we have the upstream GATSI non-methylated, and the proximal GATSI is fully methylated. In this state, what happens is the PAP-B protein, which is the first gene in this operon, is a positive feedback regulator because it increases PAP-I transcription. PAP-I, we're going to talk about right now. What does PAP-I do? PAP-I is going to be required to move the state from off to on. And the way it does that is to increase the affinity of LRP for the distal sites, which are right here. So these sites are not the same as these sites, and PAP-I has the ability to increase the affinity of LRP for these sites. These little arrows show a phenomena we still don't understand. When LRP binds to the proximal sites, it somehow causes a mutual exclusion to decrease any further affinity of LRP for these sites. So that's one thing that keeps LRP in the off state. The other is that once this site is methylated by DNA adenine methylase, this methylation also inhibits binding of LRP to this set of sites by about 20-fold. So in the end, you have mutual exclusion caused probably by changing the bending. This protein bends DNA and probably alters these binding sites. When you move, with PAP-I's help, to the upstream set of sites, you now do mutual exclusion back on the proximal sites, and methylation of these sites inhibits binding of PAP-I LRP to these sites. So it's a fairly logical switch, but we still haven't explained how LRP is going to move from this set of sites to this. If this is stable, how do you destabilize it? And here's how we know that works. We have what we talked about with somehow movement of PAP-I LRP to the upstream set of sites, but it's going to require DNA replication. And DNA replication is going to generate two types of sites. The top strand, when it's replicated, will have methylation on the top strand, no methylation on the bottom. We call that hemi-T for hemi-top. Top is methylated. And it creates a hemi-B for bottom is methylated. It turns out that PAP-I LRP can bind to a hemi-methylated GATSI site. It can't bind very well to a fully methylated, which is why even if you have PAP-I, it can't really move LRP to a fully methylated site. But it can move LRP to a hemi-methylated site. I want you to also notice that these two sites are not the same. Yes, they're both hemi-methylated, but this is methylated on the top strand. This is methylated on the bottom. It turns out, and I'll show you towards the end of the talk, that PAP-I LRP has a much higher affinity of binding to this set of sites with hemi-T than it does hemi-B, which causes the cells to have a switch bias, so the two daughter cells, one of these is going to go to one daughter cell, because remember this is DNA replication. This is going to go to another, and now we know that E. coli differentiates. One daughter cell does not have the same probability of switching as the other, and we've just shown that. We have more work to do, but we recently showed that in the lab. So in the end, you've got PAP-I LRP binding shown here to a hemi-T site, and now we activate the switch. So you can see it's getting a little more complicated. I'm going to add one more complication. Norbert Reich's group, who I haven't mentioned, is a chemist at UCSB. He's an enzymologist, and he's interested in DNA adenine methylase as well as other methylases. Norbert showed that when DAM comes in, DAM normally, let's give an example here. DAM will normally recognize a GATSI site and methylate both top and bottom strands. And normally it's highly processive. So DAM, once it methylates one set of sites, will stay on the DNA and methylate the next GATSIs very much more than it would some other GATSI far away. So it's highly processive. Norbert found that the sequences around our GATSI sites are such that DAM doesn't just recognize GATSI, it recognizes sequences around GATSI and no longer is processive. It now becomes a distributive methylase where it tends to fall off before it methylates again, and we think that may be important in this switch because you'll notice that we always have one site non-methylated and one set of sites methylated. If it were highly processive, it might tend to methylate both sites, which would not be good for the switch. And in fact, Norbert has mutants that are no longer processive, and they actually don't switch very well. So we now have some evidence that the enzymology of DAM is more complicated than we first believed, and that's due to Norbert's work on the chemistry of DAM. So now, with the modelers tried to get into the system and model all the aspects that we have, and we had a lot of aspects. For example, we had off rates and on rates of LRP and LRP with PAP-I. We have DAM methylation rates. We've got a lot of data. And they started modeling that data and were able to do it very well. For example, LRP concentration versus the percent bound. We can do this by DNA gel shift, and there's the model, and there's the experimental. Sometimes I don't even believe these guys that they can model our data this well, but they do. And by and large, it's been pretty amazing. Just using fairly straightforward formulas here for binding and putting in those the data that we have. And we found that we had a problem from the biological end. So I said initially that we didn't quite trust the modelers. Well, sometimes the modelers don't trust us, and they shouldn't. Sometimes the biologist doesn't give them good data. So we found that to get good predictions of dose responses, for example, if we have increasing amounts of LRP and we look at the probability of switching, could they predict that? So we need to get the data, and they need to find out, can they predict it? If you really understand our system well, you should be able to tell me, if I have 10 times the level of LRP in a normal cell, what's going to happen to switch rates? The switch rate is what we're interested in. How many cells switch from off to on per cell per generation? Could you tell me what would happen if I reduce the affinity of Pap by LRP for the distal site by 3-fold? What would happen? I can't tell you, and I've studied this thing for 20 years. It's too complicated. So we need to get good data. And to generate this good data, we thought this would be a cinch. We thought we'd go in the lab, and it wouldn't be a big deal. We could just take the typical way you do this is you take a lac promoter, and you take a regulator like LRP, and we would increase the level of IPTG, which induces, and we get an increase in the level of whatever regulatory protein, and we could do a dose response. There's a problem. The problem is that all these sugar operons, like the lactose promoter, have feedbacks, and some of the feedbacks we don't even understand very well even right now. And let me give you an example. If we take a GFP, this is green fluorescent protein, a short-lived GFP. We want a short-lived one so we can really look at what's happening with the population. And we simply add differing amounts of IPTG. So there's 10 micromolar, 100, and 1,000. At the low level of IPTG, where very few cells are turned on, we see two populations of cells, which is very, very bad for us, because there shouldn't be two populations of cells. We want a uniform population of cells. This means some cells expressed a lot of GFP, some expressed almost none. Here's why. If you have a few molecules of IPTG, which is the inducer of this lac, of the lac operon, if this particular cell happens to take up, which this particular cell only has a few permeases and takes up one, that cell feeds back and now makes a lot of permeases and takes up almost all the lactose, whereas the poor cells that didn't take up, stochastically take up any IPTG, don't make any. So we have cells that are just loaded with IPTG and they're making lots of GFP, and we have cells that do not. And that's not acceptable, because now we have two populations of cells. And this has been known. Various people like Kiesling have looked at this and tried to solve the problem, but didn't solve the problem, even though you won't find that in the literature unless you look at John Cronin's paper in PNES. And that was about 2003. And John showed that if you use a short-lived GFP, even the system that Kiesling developed has got two modes, one high, one low expressor. So what Cronin did, and the reason that Kiesling didn't realize that is he used a long-lived GFP. If you use a short-lived GFP, you see there's two populations. So Cronin developed a different kind of way to take up inducer. And this is really amazing the way it works. He took a LAC-Y mutant that had originally been isolated at UCLA called LAC-Y A177C, which is an altered permease which unbelievably takes up arabinose. So it's a lactose transporter that takes up arabinose, and it takes it up totally diffusively without energy, doesn't require the proton motive force, which normally is required by the LAC permease. And now we find, this is Brooke Trin's data in my lab, using a short-lived GFP, let's take a look at low levels of arabinose. So here's low level, which is where you'd start seeing a problem. And you see that you start seeing this shoulder here with increasing levels, and this is increasing time of induction. At a mid-level, you can see a nice population that's moved from here to here to here. And then at high levels, we see it moving farther. So this makes a nice uniform distribution of GFP expressed by these cells, and this is the system we've used for everything that we've given the engineers. And the second thing is you've got to figure out at these different levels of inducer how much of each of the regulatory proteins you have. And for that, we figured that fluorescence, we've tried various ways of quantitating. Fluorescence is the only way that we found that's very linear, and that would be a Lycor system. So the engineers used Markov chain modeling, and these are two papers, one fairly recently, and we now have a paper in the works to do the biology we're in the process of writing up, but haven't submitted yet, to look at the Pap switch especially. And they developed new methods which I do not profess to understand at all, even after many, many, many hours of talking to them. So I'm not even going to try. I don't want to embarrass myself. But I will give you a little bit, sort of the view I would have of how they did it. They took apart the system into all the types of reactions that could occur. Here's LRP. It can bind to the proximal sites. It can bind to the distal sites. It can bind to all sites. Maybe it doesn't bind to any. And by looking at these various states that are on, for example, here's LRP bound to the upstream set of sites. We know that's on. That's what I've talked about with you. And if they're bound to both sets of sites, that's off because we're going to block transcription. So by going through all of these states, and here's RNA polymerase, in the end, we want to look at transcription. So what they were able to do is look at these networks, these loops, and look at, for example, how PAP-I affects each of these states. For example, PAP-I helps LRP move from the proximal to the distal. We've talked about that. And so by putting all these reactions into a model, they're able to model how PAP-I works. So PAP-I stabilization of the LRP DNA complex depends upon the specific DNA location, here, or here, and the methylation, I said, affects the binding of PAP-I and LRP. So that's logical. When you come to the DAM reactions, and I say the DAM reactions, there is a lot of complexity. So take a look at this. We have all kinds of possibilities of where methyl groups can be put. Each of these green ovals represents a methylation. And so you've got all these 16 different possibilities, and methylation is irreversible. We don't find demethylases in bacteria. And now when you take a look at all the methylation states, some of these states can lead to the on state and others not directly. And by looking at all these patterns, and putting the actual interactions between one state and another, they're able to look at these as probabilistic events. And Mustafa and Brian developed a new theory and algorithms using this system that have been very, very useful for our system and others. Now, this is data we haven't published, and we're just getting ready to publish or submit. And here's an example. We give them the data, which are the data points you can see here, and we do in triplicate, and we do at least 2 or 3 experiments to get this data. As you increase the level of LRP, you have higher switching, and then it decreases. We could never have predicted that, but that's what the actual data shows, and the blue line represents their model, very closely approximating the data. What about DAM methylase? At very low DAM, we have low switching, and at high DAM, we have lower switching. And so we have a curve like this, and they're approximating our data points very accurately. And most recently, they predicted that PAP-I would just have an increase and no decrease, and that's exactly what we see. I'm not showing the data, but Brooke Trin just got that data. So far, they've been able to really match the dose responses that we've generated. And I mentioned switch bias, and we've got the Hemi-T and the Hemi-B states. And if you look at the affinity of PAP-I and LRP for fully methylated, this is KD. So the higher the KD, the worse the affinity. So these are, I told you that these methylation of a GATSI site really prevents the binding of LRP. Non-methylated site is much higher. What about the Hemi sites? Hemi-T with PAP-I is a much higher affinity than Hemi-B. And that's why we believe there is a switch bias, and now we've tested that preliminarily. Aaron Hernday in the lab started that work, and there is a switch bias towards the Hemi-T daughter. The daughter cell that gets Hemi-T switches at a higher rate than Hemi-B. And here's why. When you have this is the off state, right? LRP's bound here, protecting the GATSIs. This is fully methylated. Semi-conservative replication, the new strand's not methylated, and so we have 2 types of methylation, Hemi-T, Hemi-B. And Pap LRP, I showed you, binds with higher affinity to Hemi-T than the Hemi-B, and now we have switch bias. And now the modelers are trying to model that. So, summary and conclusions. Pap system switches randomly between on and off states, as I talked about, and they're stabilized with positive feedback. That's Pap-B that I mentioned initially. And epigenetic control is through methylation, which affects the binding of the proteins. The bottom-up model of the Pap system explicitly considers the discrete stochastic nature of gene regulatory molecules. It is a stochastic system. Multiple generations in the passage of epigenetic information. I mentioned that DNA replication is required to switch. And they are also looking at our flow cytometry data, and I do not understand how they do this. And it amazes me that they can actually look at the flow cytometry data and get more out of it than just I would by looking at just a nice peak. But they were able to actually go back to their first principles and be able to use that data to understand our system, and it appears to be working. The model captures the qualitative and quantitative effects of varying LRP, DAM, and PAP-I. The next step is to give them our mutants. We have about 300 different mutants that alter each of the steps of these, and each of the mutants has different switch frequencies, and we can ask them to make predictions, and that'll tell us if they really understand the model at the level that we need to be useful for biologists. Applications to industry for all this, predicting complex drug interactions and regulatory pathways. At least we're starting to understand a simple system, and you can see it's not that simple. Norbert Reich is working on regulatory mutants of DAM, as well as drugs that inhibit DAM. Development of vaccines against infectious diseases. The Mahan Lab is really working on this now, primarily. And DAM mutants are non-infectious, and there's a lot to be learned in the area of infectious diseases. Using sensitivity analysis to discover the most likely rate-limiting steps for drug design and targeting. And from the infectious disease standpoint as well, many pillion adhesins that are required for pathogenesis use systems just like the one I showed you. And I also mentioned that methylation is important in cancer and brain activity, and they're regulated epigenetically by DNA methylation. And I want to thank Mustafa Kamash and Brian Munsky primarily because they are the ones. There's Mustafa. Believe it or not, that's Brian. That's the picture he gave me. Yeah, he really does have long hair, and I can't see his face there. Brooke Trin is working in my lab. She's primarily got all the data that she's fed to Mustafa and Brian. Aaron Hernday is now at UCSF. And thank you very much.